Let us study about the ribonucleic acid or the RNA. Besides DNA, RNA is another important nucleic acid seen both in eukaryotic and prokaryotic organisms. In some viruses, only RNA is seen, and this acts as a genetic material. Such RNA is called the genetic RNA. Example, the tobacco mosaic virus. In tobacco mosaic virus, the RNA is helically coiled, forming the central hollow structure around which the capsomeres are arranged. This RNA is the genetic RNA because it carries the genetic information of this virus. It has a dual function. It has both the functions of synthesizing its own self and also synthesizing the proteins for the viral particle. In some organisms where DNA is present, there the RNA is called the non-genetic RNA. They depend on the DNA for the synthesis and help in protein synthesis. That means the non-genetic RNA is formed from the DNA. This is non-genetic RNA which is synthesized from the DNA, which in turn helps in protein synthesis. The non-genetic RNA is found in the cytoplasm and in the nucleus. In cytoplasm, it is usually associated with the ribosomes. RNA is also found in mitochondria and in chloroplast. These two cell organelles are autonomous in their nature. That is, they have their own protein synthesis mechanism. They have their DNA and their RNA. Let us now see the general structure of RNA. RNA is an unbranched polynucleotide chain. That is, usually it is seen as long chain. Sometimes it coils on itself. It has phosphodiester bonds between the ribonucleotide. That is, the sugar phosphate bond is same as that which is seen in the DNA. The ribonucleotide ranges from 75 to many thousands. RNA has a ribose sugar in the sugar phosphate backbone. Suppose we draw the sugar phosphate backbone like this. This is the ribose sugar. This is 1', 2', 3', 4' and 5'. At the second position in the ribose sugar we have OH, whereas in deoxyribose sugar in the second prime position only H is. That is why this is deoxyribose sugar which is seen in DNA, and this is the ribose sugar which is seen in RNA. And there is also the phosphodiester bond is seen both in DNA and RNA, but there is a difference in the sugar. One is deoxyribose sugar and the other one is the ribose sugar. The nitrogen bases are the same, like it has adenine, guanine, cytosine, but instead of thiamine, RNA has uracil. DNA shows thiamine, instead of that, in RNA we find uracil. These nitrogen bases do not form complementary base pairing. So, they do not exist in 1:1 ratio. Like in DNA, purines and pyrimidines exist in 1:1 ratio because their complementary base pairing is seen in DNA. But in RNA, there is no pairing, no helical structures, no coiling, so there is no complementary base pairing. That is why there is no 1:1 ratio in purines and pyrimidines. There are 3 types of RNAs which are non-genetic, or we can say non-genetic RNA has 3 different types or shows 3 different types. They are mRNA, rRNA and tRNA. Now let us study the structure of each of these RNAs separately. This is the diagram which is showing a cell. Within the cell there is a DNA seen which is forming the RNA. The RNA is coming out into the cytoplasm and the protein synthesis mechanism is seen. Let us first study about the mRNA. mRNA stands for messenger RNA. This mRNA carries genetic information. It carries the genetic information from DNA to RNA, from RNA to protein synthesis. That is, this is the central dogma of protein synthesis. The information which is present on the DNA is transcribed onto the RNA, that is the messenger RNA, and from the messenger RNA the information goes to the cytoplasm. On the ribosomes, the proteins are synthesized. This happens within a cell. If we draw the structure, try to explain it like this. Within the cell, let us draw a plant cell. This is the cell wall, then this is the membrane. Within the nucleus, this is an enlarged diagram here. Within the nucleus, we find the DNA, which is double helix in its nature. Let us draw this outside. The DNA is helically coiled structure like this, spirally coiled structure like that. During the process of synthesis of RNA, the DNA unwinds, opens up. One of the strands of the DNA acts as a template. This template carries the genetic information. On this template, a new strand is synthesized in the presence of an enzyme called RNA polymerase. This strand is the mRNA strand, which has complementary base pairing as that of the DNA, that is adenine, guanine, thymine, cytosine. So A always will have thymine, but in RNA thymine is not there, so uracil, cytosine, and then thymine, adenine and guanine. Like this, there is complementary base pairing. This RNA is the mRNA which is formed. Because it carries information, message, or it carries information from the DNA for protein synthesis, this is called the messenger RNA. And this name, messenger RNA, was given by Jacob and Monod in the year 1961. It has a molecular weight of 5 into 10 to the power of 6 daltons, and it constitutes to 5 to 10% of the total RNA present within the cell. It means 5 to 10% of the RNA which is seen in the cell is mRNA. After the mRNA is formed within the nucleus, the mRNA slips out. Here you can see very clearly, this is the DNA strand on which the mRNA is formed. This mRNA strand slips out into the cytoplasm, where the ribosomes bind to the mRNA strand and they help in the process of protein synthesis. In prokaryotic organisms, in prokaryotes, mRNA is metabolically unstable. That is, it has a very short half-life period of around 2 seconds to 2 minutes. And this very short half-life period will destroy the mRNA within this time. So, in prokaryotic organisms, when the mRNA strand is being synthesized, that is, during the process of transcription, or the process of transcription and the process of translation occur simultaneously. What does this mean? That is, as the mRNA strand is being formed, this is also decoded for protein synthesis. That is, formation of mRNA and formation of proteins occur side by side, because this is very short-lived in prokaryotic organisms. In eukaryotic organisms, it is stable when compared to the prokaryotes. The half-life is from few hours to few days. So it doesn't get destroyed immediately. It remains for some time and translation occurs one after the other. That is, after transcription, translation occurs. Both in prokaryotic and eukaryotic organisms, the RNA strands have a 5' leader sequence, and there is a central coding region, and then it ends with a non-coding 3' region. That is, there are 3 distinct regions, like you have a 5' region, then there is a central coding region, then there is a 3' region. Now let us see what is the difference between a prokaryotic and a eukaryotic mRNA. Let us take a new screen. We will see the difference of a prokaryotic and a eukaryotic RNA. Prokaryotic mRNA. In prokaryotic mRNA, the genes which are concerned with the same trait are present in clustered groups. If this is the mRNA which is being formed, then the genes are present clustered. That is, the operon groups are present together. That is, the structural and the functional genes are present together, as a result of which large number of polypeptides are formed from the same RNA. From this RNA, many proteins, large number of polypeptides is many proteins are formed. And this is why this mRNA of prokaryotes is Polycystronic Mrna, because it is producing large number of proteins. It is polycystronic in its nature, and we call it as Polycystronic Mrna. But this mRNA does not have a cap. There is no cap on the mRNA on the 5' end. And because there is no cap on the 5' end, the ribosomes bind randomly at many positions on the mRNA strand. This is the reason for synthesis of polypeptides, multiple or several polypeptides. If the ribosomes have a specific position of binding, then protein synthesis will start only from that place. But because the ribosomes are bound randomly along the length of the RNA, many proteins can be synthesized. Now let us see what is a eukaryotic mRNA looks like. How does it look like? The eukaryotic mRNA has certain special features. It has a cap at the 5' end. Let us draw it like this. It is a single stranded structure, usually a long chain with a number of codons present on it throughout its length. It has a cap. If this is the 5' end, this is the 3' end. On the 5' end, there is a cap. This cap is methylated guanine. Methylated guanine. And this caps gives protection to the mRNA from the exonuclease enzymes. That is, when the mRNA comes out into the cytoplasm, there are all chances of this mRNA being broken down by exonuclease enzymes. That is, they destroy this mRNA. To prevent that destruction, the mRNA comes out in a disguise with a cap, and that cap is here, the 5' and the methylated guanine is added as a cap. Immediately after the cap, there is a region. This region is called the non-coding region, or we can call it as the NCR region, the non-coding region. This region has around 10 to 100 nucleotides, and it is rich in adenine and uracil, and it does not translate. There is no translation occurring, that is, there is no protein synthesis, no information for protein synthesis in this region, and this is called the leader sequence. Immediately after the leader sequence, there is a codon. This region, there is a codon. This codon is called the initiation codon. Initiation codon in both prokaryotic and eukaryotic organisms is AUG, which is for methionine or formyl methionine. Beyond the initiation codon, the region is called the coding region, which is represented by CR. CR stands for coding region. This coding region has about 1500 nucleotides. And the protein synthesis occurs in this region, that is, translation or protein synthesis occurs in this region. After the coding region, there is another region which is called the termination codon. There is a codon here which is called the termination codon. The termination codon is also called the nonsense codon. The reason for being calling as nonsense codon is this codon does not give information for any amino acid, and so the protein synthesis ends here. When the ribosomes come across this nonsense codon or the termination codon, the protein synthesis is stopped. The termination codons, I'm writing in short, the termination codons are UAA, UAG and UGA. These three are the termination codons. After the termination codon, the mRNA strand has a long tail, and this tail of mRNA is called the polyadenylated tail. Polyadenylated tail. That is, it has multiple adenines. It ends like this, adenine. And these changes in the mRNA of eukaryotic organisms, all these changes, that addition of cap, non-coding sequence, and the addition of tail, cap and tail, occurs after transcription. After transcription, that is, after the formation of mRNA, the cap and the tail is added. Only when the cap and the tail is added, the mRNA comes out from the nucleus into the cytoplasm. Now let us see the next type of RNA. This RNA is the ribosomal RNA or rRNA. Ribosomal RNA or rRNA. It is also called the insoluble RNA. Ribosomal RNA or the insoluble RNA. Within the cell, it constitutes to 80% of the total cellular RNA. Of all the RNA, 80% is rRNA. It is found primarily in the ribosomes. It has a single strand. Of nucleotides, but it forms pseudohelix. At several places. That means it coils over itself and then forms pseudohelix. And when it forms pseudohelix, base pairing occurs. More than 50% of this RNA contains the GC content, guanine cytosine content. That is, in this complete RNA, more of G and C is present. In prokaryotes, the rRNA is 70S type of rRNA is seen, or 70S type of ribosomes are seen because it is basically concentrated in the ribosomes. In prokaryotic organisms, we have 70S type of ribosomes, which has 60% of RNA and the remaining 40% is the proteins. And this 70S RNA is further divided into two subunits. That is a smaller subunit which is 30S subunit and a larger subunit which is 50S subunit. The 30S subunit has 116S rRNA and it has around 21 different types of proteins. That is, the smaller subunit it is made up of 16S rRNA and 21 different proteins. In eukaryotic organisms, the type of ribosomes is 80S type, where it has 40% of rRNA and the remaining 60% is proteins. And in this, it has a smaller subunit which is 40S subunit and the larger one is 60S subunit. The 40S subunit in turn has 28S, 5S, 58S and 5S rRNAs. Plus it has 49 different proteins. This is, and the structure is, the ribosomes are formed. Let us see how the ribosomes are formed. The ribosomes usually have, now we have seen that there are two different parts. One is RNA and the proteins. The ribosomes have a central core of protein around which the RNA is wound. Here we can see the diagram. There is sugar phosphate backbone This is a single strand, but because of the pseudo helix, a false helix which is formed, there is base pairing seen, complementary base pairing seen between the two different nitrogen bases. Now let us talk about the next RNA, that is the transfer RNA. This is a cloverleaf model of transfer RNA, but first let us see what the general structure of transfer RNA, what is present in transfer RNA, then we'll discuss about the model. This is transfer RNA, or in short we can write tRNA. It's also called the soluble RNA or the adapter RNA. Within the cell, it constitutes to 10 to 15% of the total cellular RNA. That is, of the total RNA, it is 10 to 15%. It is the smallest and is composed of 75 to 95 nucleotides. It has number of unusual bases, like it has a DHU unit or a base or a loop. DHU stands for Dihydrouridine. Dihydrouridine. This is one loop. Then you have another loop on this which is called the psi loop. That is the psi loop. Then there is another one which is called the inosine. There are certain peculiarities on this structure. And then there is methylated bases and thiamine. These unusual bases are produced after transcription. That means after the tRNA is formed, then all these changes are seen. That is a modification of the normal nitrogen bases, adenine, thiamine, cytosine and guanine. The normal ones are being modified. It shows helical structure, considerable helical structure, and more than 50% bases are paired. Pairing is seen. You can see in this structure there is pairing here. In the arm there is pairing, in the loop there is no pairing. 50% of this RNA is paired. The model of tRNA, this particular model of tRNA was proposed by Hole, which is called the clover leaf model. This clover leaf model supports its structural and functional properties. Any model which is proposed should support the structure and the function of that particular structure. According to him, a single polynucleotide chain is folded backwards on itself and results in the formation of getting closer of 5' and 3', because the single strand which was like this, it got folded, and then which resulted in the closeness of 5' and 3', which results in the coming together of 3', 5' ends. And this folding resulted in the formation of 3 major arms. There are 3 major arms, 1, 2, 3 major arms. Each arm has a stem. This is the stem and the loop. The loop has unpaired bases. There is no pairing here, whereas the stem has paired bases. Now let us see what is the uniqueness of this structure, of this model. All the tRNA molecules contain the same sequence, terminal sequence, CCA terminal sequence at the 3' end. And this ends with adenine, which is the last residue at the 3' end. This site is the site for the attachment of amino acid. Amino acid attachment site. This is the amino acid attachment site. Then there is another loop which is called the T-Cy-C loop, or this is called the thiamine arm. Thiamine arm. It is located near the 3' region, very close to the 3' end. The thiamine arm is. It has 7 unpaired bases. In this loop, there are 7 unpaired bases. The T-Cy-C arm is involved in binding to the tRNA molecule to the ribosomes. This tRNA molecule, in this place, it binds to the ribosomes. Why should it bind to the ribosomes? During the process of protein synthesis, the tRNA goes to the site of protein synthesis. It decodes the information by this anticodon region, and then it has to bind to the ribosomes to understand the coding. So this place it goes and binds, this region binds to the ribosomes. Then there is another loop which is called the DHU loop. DHU loop stands for Dihydrouridine loop. This is made up of 8 to 12 base pairs. These 8 to 12 unpaired bases. Not paired, unpaired bases. It is the recognition site of amino acyl synthetase. This region recognizes the amino acyl synthetase enzyme. Then there is another region which is called as the Anticodon Arm. This is the Anticodon Arm. In this Anticodon Arm, there is one nucleotide triplet. We find the nucleotide triplet here. All the tRNA molecules have this site, which is the recognition site of the complementary triplet codon. On the mRNA, there is a triplet codon. This codon cannot be understood by anybody. Only this anticodon can decode this codon. So, when the tRNA comes to mRNA, then this anticodon will understand this codon by this anticodon site. Some tRNAs which are very long, they have an extra short arm. This is the lump. tRNA helps in bringing the amino acids to the ribosomes and plays a very important role in protein synthesis. See, during protein synthesis, the mRNA which carries the genetic information, 3 codons will code for 1 amino acids. Amino acids are in somewhere within the cell. The amino acids are somewhere in amino acid pool. These amino acids have to be brought from that part to this region for proteins, for decoding and for protein synthesis. So the bringing of these amino acids from the amino acid pool is done by the tRNAs. That is why they are called the transfer RNAs. So there are around 20 amino acids, but the number of tRNAs are 32 or more. That means there is more than one tRNA to carry one amino acid. There is no 1:1 amino acid and the tRNA. tRNAs are more when compared to amino acids. That means more than one tRNA can carry one amino acid. That means the anticodon region in 2 to 3 tRNAs is same for some particular amino acid. Now these are the 3 different types of RNAs, their structure and their function. These are some questions which you can try to answer for this particular topic.